Very good evening to everyone. It's my again great to meet you all once again for a very interesting topic in this evening. I hope everyone will be more curious to know about how we are going to bridge a gene expression and the chemistry. So far, many webinars and many sections have told about what is the gene expression and how to do the gene expression analysis. And after knowing about the gene expression, how to take up that process. And this is something different. We want to introduce to you all how to connect your. See, there are lots of gene expression studies have been there, and from the gene expressions, how we are going to connect your chemistry. So, chemistry in the sense we are going to concentrate on drug discovery and development. That is, we are going to connect the two strings, two end of the strings coming to our same platform. Let us, I cannot wait to start the sessions. Let's go ahead. I am Divya Shanmugarajan here from Altum Technologies. We are going to present about how the bridging of a gene expression and chemistry would benefit for various research applications. I am working as a senior application scientist for the BIOVIA Discovery Studio National Technical Support. Let's go on. Before getting into which, I would like to introduce my company. So Altum Technologies, it's more than a decade company, had a very good imprint in a 3D printing technology, and one of the wings into the life sciences, the BioVR Discovery Studio, Material Studio, and Pipeline Pilot. And we are also into the 3D bioprinting technology for various cell line studies and so many. If you want to build a cells in 3D scaffolds and you want to test with the drugs, even we have a strong imprints and we are keep moving on this technology. So please look into our website for more details. You get more details about the life sciences as well as the 3D bioprinting technologies. And also you can see our esteemed Altam Awards, which has been awarded for us for an 2013, 14, 15, and 17, and so on. Okay. So when you want to start something, there should be some platform we'll be working on. So we are having a rich scientific portfolio of the BioVia platform where we are trying to explore the various research activities, right from the unified lab management where we are trying to replace in a paper works into an electronic laboratory notebooks. And say for instance, when you are trying with an writing us things and keeping for a record for a while, definitely after a long some time period it will try to decay. So in order to avoid all those things, we have introduced in a unified lab management techniques. Of course, process production operations, if you have any process which should need to be pipelined and need to be aggregated and you want to monitor in terms of dashboard analysis, even we have the solutions for this. Any software which you are developing or you want to use it, it should go for a quality and regulatory management. So we have a separate domain for the quality and regulation, which includes the documentation, processing, compliance, and collaborations. When I explain about a collaborative science, we have a solutions for all kinds of research applications in life sciences. Say for instance, if you want to predict the models, if you have a sequence, you want to know about its dynamic pattern or the what is the sequence analysis and the physiochemical properties. Right from the sequence to chemistry, you can have the predictive model prediction analysis of them. And novel therapeutics. When I say about a novel therapeutics, immediately the big molecule or a macromolecule will come to and show. We have a very good workflow to do the antibody modeling and drug discovery process to see the antigen antibody interactions and engineering materials. So even if you discover a drug that has to be delivered properly using a various materials or an biomaterials, it could be a cell fibron or maybe carbon nanotube, which material or the drug vehicle is more appropriate for delivery. So all these things in very precisely you can study by using a material studio. And of course, whatever the drugs that we are taking or a compound that we are prescribing or we are going to discover, that should free from toxicity and mutagenicity. And also it should obey the pharmacokinetic properties. So right from when we have various animal models, which is built by using machine learning algorithms or the part of this chemicals and formulation. With this brief introduction, I'm getting into the today's topic. So, quite interestingly, everyone will like to know about the gene expressions. So, gene expression, it's a every day, even when I speak, certain genes are getting expressed. When I think, some genes are expressing. When you are hearing, some genes are getting expressed. So, gene expression is an omnipresent. To understand the nature or how it is there, it is the most important and prominent role in your drug discovery process. So that's why the gene expression will help you to understand the molecular signatures. See, what I mean by molecular signatures could be a biomarker. If the breast cancer is expressing, definitely there will be an EGFGR or estrogen receptor or some kind of a biomarker will show this is because of this gene expression is happening of certain diseases. Also, this gene expressions will help us to determine the mechanism and toxicity of the drug candidates. Once you know which gene is expressing, and if you know the mechanism, one can discover the non-toxic drug candidates. And also, it aids to profile and plays a many roles in your drug discovery process. Profiling of a genes, some genes are top regulators, some genes are down regulator. If you know this profiling, we can help. It will actually plays a major role and gives you a part in your drug discovery process. And clustering of some genes, say for instance, if the person is having a high diabetic and they have problems of obesity also, there are cluster of genes will be totally expressed together. So we can identify those expression patterns and accordingly we can come out with a drug leads to discover for an specific biomarkers and then how the drug is getting responses. As I mentioned, gene expression profiling is a one universal screening platform for any kinds of drug discovery process. And also, it will help you to understand the molecular level interactions and what is the molecular level expression of a drug associated with the phenotypic versus genotypic changes. So that's the gene expressions are very, very important to know in the to understand even a drug discovery process. So there we have a various way. How do you study this gene expression? I was keep on. So earlierly people will see the QT-PCR and then PCR analysis. All these things are earlier molecular characterization or molecular biological techniques, I could say that, are being used. Now we are getting into the advanced techniques. So that's why to study your gene expressions, they have new techniques like a whole genome sequencing analysis, RNA sequencing analysis, microbe sequencing, miRNA, metallome, chip, and microarray. Microarray is quite known for more than a 2 decade, which is the first adapted technology to understand the gene expression. So these all are the various ways where you can study the gene expression analysis. So a little bit about an RNA because uh, I could tell that many universities or even many studies who is working on research applications or trying to work more on RNA sequencing analysis. Why this is gaining a more importance in the sense this RNA sequencing analysis will help you to understand the transcriptomes. What are the transcriptomes? You can see here if you have a if it is a this is like a cancer, there will be heterogeneous tissues will be there. So if we want to understand the in the cell population how many RNA is expressing, how it is expressing, and having that profiling, one can able to understand which gene is get expressing more. So it is like a technique you have to understand. So RNA sequencing is gaining a more popular among the researchers. They want to study now. It has been downstream, and they are working on a more single cell RNA analysis also. So single cell RNA analysis is applicable for any kind of fields, including in the cancer, and it will help you to analyze the heterogeneity of the tumors. Say, for instance, I have described here heterogeneous tissues. One can go ahead with a bulk RNA sequencing analysis, that is totally extracting of the total RNA and sequencing it and going into the downstream analysis using an R, bioconductor, or any kind of a pipeline, and understanding the homogeneous cell population or gene expression data analysis. On the other hand, we have a single cell RNA sequencing analysis where we try to take in a single cells, individual cells. You can see here red color, we are taking a red color as one of the cells and yellow color as one of the cells, and green and cyan colors. So single cells are being isolated and it be barcoded with cDNA and sequencing and identifying the unique transcriptomes and expressions of the diverse cells. So cell to cell variability we are trying to study in a single cell RNA. So this technique will be more effective and therapeutic and help you to study the very specific targeting for a pathogenic tumor cell lines. So that is why the RNA sequencing is for gaining more popular in your various diagnostic method as well as like diseases like a cancer. So we should not forget about our microRNA. Still, even in some places, the microarray data analysis is also going on. It's a very well-known technology to understand the expression of the genes parallelly. We have thousands of genes expressing in a one single small microarray. We can able to predict the analysis. So it is a kind of an in vitro technique used to detect your expression of thousands of genes at the same time. If you have a normal cells and as well as your express cells, you can put together and you can make it with an cDNA and then mark it with an CY3 or CY5 the dyes, and then hybridization. Basically, it is a hybridization technique. And then using hybridization technique where we are trying to transform into the image. From the image, we are trying to introduce downstreaming analysis using various statistical parameters. So this technique will be known to understand the genetic and epigenetic makeup of any kind of cancer cells. And also, it will be highly parallel and high throughput genotyping applications. Even it is don't only have its applications in cancer diagnosis or any disease diagnosis, even in the plant, even the whole genome sequencing, whatever I have mentioned, it is applicable for even the both eukaryotic and prokaryotic organisms. And this will have a huge application plant genetics and crop improvement also. Using this technique, you can identify the new genes are getting expressed, which gene is getting expressed. That also we can identify from that. From that, one can study the how the function and expression will vary in the different cell conditions. So depending upon the conditions, you can study its functions and how the genes, whether it's expressing more or upregulated or downregulated, that we can understand. So this is how the microarrays begin, and then from that extension, people are taken to the various next generation sequencing analysis, which I mentioned in multiple methods. So downstreaming analysis will always go like this for a gene expression data, especially for a microarray. Feature extraction, that having that cells that of output of that, you have to do the feature extraction and quality control. Any kind of analysis will definitely will go for quality control. Even it may be a RNA or maybe a DNA or methylome, whatever whole genome sequence. Pirate they will use for an NGS analysis. Here we have a different quality control methods for our in microarray data analysis. Then normalization, differential expression analysis between the two reference cells, and finally biological interpretation of results and submission, because there is a GEO databases where we have accumulation of various sequencing analysis informations, including it will show you about if you make an advanced option, you can get that which is an transcriptome array, which is an array technique or methylome or whole genome RNA. So like this, they will submit in the GEO database, will give you the more idea about how which technique they have followed and how they followed, what is the type of experiment they followed. All this analysis we can able to, through that we'll get extract our data. Okay, so if you want to do downstreaming analysis, we should know definitely the scripting languages, either R or even you should know the Python. Both the platforms have been widely used in this analysis, in a put in a simple word, is a machine learning algorithm and artificial intelligence techniques must be included, and more into when the machine learning algorithms, how they are going to use. So platform, it's generally the R with a bioconductor, or you can use it via to the R studio also. There are some online pathway, I mean pipeline databases is also available, or an pipeline is available through that also one can upload the data. But try to be in a differentiate from those analysis. We are introducing a pipeline pilot, which is a product of the Dassault systems, BioVia Dassault systems. Let us have and see how the system is. It's actually it's in a how it is going to accelerate your machine learning process of any kind of data you have. This will give you the powerful solution. Let's see this.
Science, engineering, and process-based R&D organizations are often burdened by complex, disconnected IT environments and disparate data sources. These challenges make it prohibitively difficult to analyze large volumes of data, to make predictions, to derive results, and to fully leverage data.
Especially if you have a big data analysis, pipeline pilot is the best solution.
In model development and machine learning. The shortage of data science and machine learning experts also hampers widespread model development. Deploying models across the organization can be slow, labor-intensive, and error-prone. As a result, many important insights are missed as organizations strive to innovate and generate value. Data Science for All leverages BIOVIA Pipeline Pilot to provide advanced analytics, predictive modeling, and machine learning capabilities that can accelerate R&D innovation and boost productivity. Dassault Systèmes data pipeline tools make predictive analytics accessible to researchers in any industry that analyzes large volumes of data to generate insights. Data processing and preparation is faster, easier, and more accurate, allowing teams to focus on better decisions based on better science. Data science allows researchers to detect meaningful patterns in data. These patterns can help them understand complex systems, decipher atomic and molecular behavior, optimize processes, and make new discoveries backed by knowledge. Dassault Systèmes provides the tools required to manipulate and transform data from a variety of sources. The easy-to-use interface speeds adoption while making it quick and easy to build and share powerful models. Train advanced models using the latest data science techniques without writing a single line of code. Drag and drop.
So here we no need to write any coding because all the codings which has been constructed in terms of components is just like pulling the component and running the analysis.
A single component to implement a random forest algorithm, parameterize a Bayesian classifier, or train a neural network. Dassault Systèmes technology makes powerful machine learning algorithms accessible to the entire team, regardless of skill levels. Data science tools from Dassault Systèmes facilitate chemical search and similarity analyses, formulation development, genomic analysis, and the building of sophisticated machine learning models, from k-means clustering to convolution neural networks. The technology also handles data visualization and reporting. By simplifying the creation of powerful machine learning algorithms, Dassault Systèmes data science technology promotes best practices that can accelerate collaborative innovation with powerful predictive models and workflows. Contact Dassault Systèmes for more information about data science.
So hope this is the pipeline. See, when you want to write up codings and you want to see the results, definitely, even if you miss single comma or even if you miss a single line or a name, case sensitive, even every words are case sensitive. If you're going to miss it, definitely it is going to make your work hazel. At the point of time, people will like to hate the scripting garbage. But the Dassault systems based a pipeline palette solution will help you to resolve the problems, just pulling the component and running the results and seeing out how effectively you can run it. You no need to have expertise into it. If you want to develop, if you already know the scripting, you can develop your own protocol in a pipeline pilot. So with this technique, I'm going to show how the interface of pipeline pilot will be there. This is how the interface of pipeline pilot will be there. So it will be like a full of components where we have, and this is a pipeline pilot client where we have those options and then protocols and the components. Basically, we have more than 3,000 components for the machine learning algorithms, including chemistry, biology, imaging, documentation, materials, and life sciences solutions. So we have where you want to find it out, a basic statistical analysis also can be easily done by using in a search menu just by typing a simple words, keywords. You can grab that specific component and you can connect a pipeline like this. So this is a protocol window where you are trying to develop the new protocol for your name, or you want to develop a new workflow for your work and for your analysis. And the component you can pick, which will be already inbuilt within a scripting languages. You no need to try new. If you want to make a changes, you can make a changes in your parameters wise, and you can use the same. And we have a status bar and the task bar, and you can see the jobs when it's running and the help options. If you have made some error in your pipeline, it will help you to fix out of where the error is and why the error is. It will try to show where the problem is arising. So you can easily fix that using a help menu. And you can also watch when you're running your protocol where it is running, where it is stuck also. So such an interface will help you to drive you a downstreaming analysis of your process very easily. The execution of the process, if you have more than one protocol need to be ran in parallel, you can use the protocol executions using aggregating the components. So it always starts from a left to right, say taking from the text, customizing and goes on, and then from a top to bottom. One can have a multiple protocols in the same pages also. That means if you want to run parallelly or need to be continuous, all these things can be taken in account while executing your protocols. This is how the protocol execution will happen. And types of components. We have, as I mentioned, we have more than 3,000 components are they right from a viewer. If you want to view your results in table form or Excel files, or you wanted to you view it in a PDF format, so whatever the format that you're looking for, or I wanted a HTML format, HTML5 format, whatever you're looking for, you can make it in a viewer window. And expressions, that means, say for instance, I want only this expression has to be satisfied. I don't want second expression should be not be satisfied. So if you have a kind of an expression, A should be equal to B, C should be equal to B. So that's kind of a condition-based expressions can also be done by using an expression components. And you have a sub-protocol components. That means if you're building a very big pipeline and you don't want to show it in a window, you can just merge all together in a component. It's called sub-protocol. The sub-protocol will have a passport and then a fail port. If it is, say, if you're screening a compounds or gene expressions for the specific pools, it will try to take up only the specific things in a passports, and what are the fails will be comes out of the fail ports. And we have a generator, filter menus, readers, calculators, writer, joining, and the various components. One can edit the various components. So these are the some of the components we have. However, a pipeline, but there are still more components out there. As I keep walking on, you can see even those component options. So now I am trying to stick towards the technology of a microarray, because today's session is going to be like from the microarray data analysis only we are going to connect our pipeline. So if you want to see that, it should start with a feature extraction. So after just imagine that you are done with your microarray data analysis, you have a output like this, which is in a form of cell or in a text format. So we have a different platforms will be easily accessible in pipeline pilot. That is what I have mentioned here. So Affymetrix platform, Elegant platform, GenePix platform, Illumina platform, Nimble 4 platforms. So all these platforms input can be easily read. Even if you have an Excel sheet formats, if you have an Excel formats also, you can take it. Or you have it download from a GEO query also. You can automatically download from a GEO query as a soft file, or you can fetch it data from it. If you know that the GEO number, you can fetch it directly from a GEO data fetcher also. So these are all the typical formats should be satisfied. So if you have .cl for Affymetrix, and it will try to analyze an R packages, FE, Lima, Oligo, and then if you have for an elegant, R packages, including lemma. So we have a specific packages which is already inbuilt into it, and only at the beginning you have to locate your R files. That's it over. Once you locate globally your R path, then you can automatically take up and run your process. So first is a feature extraction. That was by trying to read our data from an affy matrix. Once you read your data, so this is how we have to read your data. So take your data, read your and truncate your experiment if you want to truncate with some levels, and then view your results. So our source will be in the text formats and background corrections, normalization method, and then summarization. So when you want to run in terms of a course, each and every line you have to write it, and you have to say all this. This is a background correction. This is normalization. Here, all it comes in terms of options. Just selecting, so that means for each and every protocol which you want to run will take some amount of time to run it in your scripting languages. But however, you can run parallelly any number of results in pipeline pilot. That's how your results will be. You can run parallelly. Also, it will be have more efficient and accelerate your process. And once you read it, that's how the quality control will be. So quality control will making inspection of your data, of your scanned images, whether how the hybridization is quality is there, how the RNA quality is there, how the statistical quality is there. So we'll try to use an histogram-based analysis, and also we can predict the RNA, how much quantity of RNA is left out and which present in the source. That also can be taken in terms of an histogram data of a various samples. So that's why we say that histograms are very good visualization tool for identifying the saturations. So this is how they check a quality control in this. Moving on, this is also one of the method to understand whether the how the intensity or the how the data has been distributed. So box plot is again one of the good visualization technique to understand the intensity of all the probes across all the arrays, and this has been drawn from a 25th to 75th of percentiles, or even you can draw for a median or 50th percentile, and to see the how the spread of data. So all the array must be in similar range, otherwise it should be discarded. Say for instance, you have some array over here, some array over here. So it should have all the data must be a good array. One is now over here, one is over here. So that is not that will not be considered a bad array. Either you have to optimize, otherwise you have to take out of the box. Unless we should not take out of the box without any reasons. That is also very important in your, any kind of a gene expression data analysis. You should not delete any data. If you are taking out any data, there should be a perfect reason must be stated why you are not taking a data. What is the reason behind it? Or you have to make a multiple option based parameter optimization technique. Then only you have to take out the data from your analysis. So make sure these are the kind of quality control analysis will help you to sort your data. So background corrections. So goal of the microarray is in the based on intensity value, which has been considered as in a proportion of a level of expressions. So based on that, we have an various background control data. So including you can see your RMA2 method, and that is a robust multi-array averaging method. And we have MAS methods, and we have MASI methods, and GCRMA method. That is a gene chip RMA method. So all these methods will be taken, and this will show this based on the how much hybridization has been produced in the samples and targets, and also it eliminates the signals of a non-specific binding also. So this is a background correction, which is most important part of your experimentation. And coming to the normalization, so it is also used to control the variations between the assays, and then how many ways to normalize array type or design of experimentations or assumptions. So for expression atlas, for example, affymetrics microdata are there. Usually people will go for RMA method with an oligo packages. It's all the packages which is present in the R and bioconductor will help you to drive this process. And of course, if you have an elegant microarray, they will use the lemma packages. So normalization will be like this. We have an MA plots and box plots will help you to understand and remove the aberrations and outliners of a non-biological factors. And relevant patterns and outliners of the actual biological factors alone will be taken together. So normalization or needed to be accurately interpreted with results. And you can see the results before normalization, after normalization. You can see the variations in the results. So all are same together and arrays are perfect and MA plot is also good. And if you have to analyze an gene expression data analysis, definitely differential expression analysis is the most important one. The main aim of is to identify which gene is expressing under which condition. If you have two different samples, maybe you're having a cancer or maybe in a normal sample, you want to see the differentiation, which gene is expressing more as compared to the normal samples. That can be done by using a differential expression analysis. An important consultation is in considering a multiple testing files. And this, moreover, there are the statistical parameters will help you to compare the thousands of data and a same comparison data you can able to get retrieve the multiple gene expressing in the multiple conditions in a small number of samples also. Some microarray experiments will have a less than 5 replicates per condition, but however, this leads to increase the chance of the false positive. So very important thing is the gene expression, differential expression analysis, which is just we are going to call here as I'm explaining over here lots of theories, but all I when I do the process, it's just an 1 or 2 minutes work. Then I pull the component, input your data, just finish it off. That's it. Coming to here. This is again a pairwise. As I mentioned, you no need to write any codes here. No need to write any scripting. Already we have a component here. Just take the component, pairwise differential expression analysis, and then we have a various method, T statistics method. We want a two-tiled or one-tiled method along with the correction method FDR, or we have Holm or a Honchberg, Hommel, Bonifero. Like this, we have a various correction methods you can select it. And the factor label, normal cell and the cancer cell. Output should be what you want, p-value less than 0.05 or less than 0.00. So like this, you can select your parameters and you can compare. And applicable for filtrations. So we are not going to take up all the results actually, right? We are going to set a threshold for the output which you are doing analysis. That's why when you have the p property values, as I mentioned earlier, we are going to keep the threshold 0.01 true and filter less than or equal to or greater than or equal to which method you wanted, pairwise differential expression. So once you keep this absolute true and run the process, you will get this. So here also, this is the next pipeline. So pairwise differential expression analysis, selecting from the property values. And finally, extract the subsets. So here in the express of subsets where we are trying to, so this is how the sub protocols will be there, box to box to box. This is actually the output will be shown here. When you double click and see that, it will have an experiment like this. When you again double click and see this biological gene expression data analysis, you have a matrix dimensions, saving the selections, non-empty sets, and how many rows, number of rows and columns matching. It's again a very big task when the people work it on a R-based programming. So missing a single row or not able to retrieve it, again they have to rerun the process and they have to match with the specific gene sets. So here you don't see such a kind of an analysis. If rows are not matching, where it is not matching also, it will try to show you a difference. So finally, after doing this analysis, then we have to go for look for heat maps and clustering, gene enrichment pathway analysis, and after identifying a specific genes, we have to see it in a David pathway or in a GCSE fee or some kind of an enrichment pathway analysis databases and the network analysis. So this is how we'll be identifying your genes. Now you are end up with the genes now. So after that, annotations. So when you isolate a gene, we have to know its gene bank, where the chromosome, at what are the PubMed IDs, what the gene ontology. Here also, it's not like that. Just to pull the component and get the results. So at the finally, when you run this protocol, you will end up with a gene analogy of what our genes are expressing in a specific gene samples. So just start, give you only dot cell input, and then look for your outputs. This is how the pipeline will be taken. And if you want to individually separate extract the individual genes, of course you can do that also. give the name of the genes and selected genes or any specific top array parameters, and you extract the individual genes. Now, as I mentioned, we have not done with the gene part now. Okay, now genes are ready. How should I proceed? I know this is a biomarker or expressing. This is the gene is expressing. If you are doing NGS analysis or a microarray data analysis or maybe RNA-seq, whatever it may be, some genes are getting upregulated in some patients. So from there, how I am going to proceed? This is a big question. So am I going to discover the drugs? Am I going to end up here and going to state this is the only gene expression? And what is the outcome or solution? So now everyone will be thinking, I identify the biomarker is expressing in a cancer patient 1 and some other biomarker in cancer patient 2, cancer patient 3, or maybe some kind of a disease like TPR gamma or whatever the disease is. How I am going to proceed? That's how we have a solution. We are going to bridge your gene expression with your chemistry. Output of a gene expression, we are going to bridge in a chemistry to understand which drug or the lead must be preferred for the specific gene which is expressed from your analysis. So that's a bridging we are going to see the pipeline now. I hope everyone will be more interested to know about it. Okay. I'm going to take only the microarray since I have taken as an example of a microarray. I'm going to stick on those pipelines. So after doing that analysis, I have a microarray data and I'm going to compare. I'm going to get from the expressed genes, the respective chemical compounds need to be isolated. Let us take the pipeline where we have a significance of leukemia genes from an experiment of annotations including many information of the microarray experiment, that is the AME annotation. It will help you to search for the signaling pathways and the compound IDs from the cake database. And finally, we are going to connect with the NIST library to take the corresponding structures and we are going to view the results. Let's see this each and every step in detail, how it is going to connect by means of various components in the pipeline pilot. So overall workflow will goes like this. Calculate the fold change. We are giving the conditions. We are telling the conditions to the protocol. How much you want, how much fold change you are looking for. Everything it is under our control. You can have a full control on your experiment on a pipeline which you want to design. If you want to make a changes, of course you can make it changes and you can run parallelly also. So calculate the fold change and you are trying to tell the which genes with a fold change greater than 2 or 3 or 1, whatever the experiment that you want to keep it, that you can decide. And after giving that specific genes of fold change and add a symbol. What is a gene symbol? It may be an PKCI3, PTN, like an PPR gamma or EGFGR. Like this, we are trying to add a gene symbol and connect with the cake pathways and annotations. And you from the cake, because cake is in the integrated a multiple network pathway where we have even the compounds also in that we can able to connect very easily. So we are trying to take from the annotations, we are connect with the pathways, and then we are going to extract the Cas numbers and retrieve the compound information, and finally going to display for which gene, which compound, it results in terms of a table format. This is what we are going to see in another 15 minutes. Fold change. Computing the fold change. Okay. Let us consider in a very easier way. You have a normal cells and some other disease cells, two factors. We are going to see the fold change between the signaling between the two different labeled parameters. So no need to do anything, just call the component fold change. Once you call the component fold change, you get a component like this. And it will ask you the factors. What is the factor 1 and factor 2? Are you going to look for a logothromic? Or are you looking for a signal to log ratio? And output property will be a fold change. And this is how it will calculate in terms of mathematical equations. Let me put in a very simpler terms. And next we are looking for a fold change. So we are trying to apply the threshold here. Which threshold you are looking for? So fold chain, it actually measures how much quantity between the virtual and subsequent measurement should happen. And this is a ratio between the two different quantities. Say for example, A is a cancer and B is a normal cell. The fold change of B with respect to A is B by A. In other words, 40 to 80 is defined as a fold change of 2. Like this, it will try to put together and gives you a change. So you can filter only these fold change results and drop down the other one. That's why we have a fold change, we have a pass and the fail ports. You are selecting the genes based on the top level numerical array property. I'm now selecting only top hit genes. Finally, after selecting it, we are using a bioconductor affy package. And here also, no need to worry about it. Just call this add probe annotation component. And what do you want to make it annotation? We have all this kind of components. Just call for location, gene ontology pathway, everything together using in a bioconductor and affy path, affy package. And then once you call, it will automatically updates with the respective genes and informations and some terms of rows and columns. So which I mentioned earlier, I'm trying to skip these files. So extract the experimental subset. Once you extract, and again it will directly goes into an get all pathway IDs and AME annotations. Once it gets into it, what and all it will do, it will try to look for the properties and try to return. This is a sub protocol. Inside this, we have this protocol actually. So take pathway ID, merge the data, sort the data, isolate the pathway ID, and merge and identify each pathways. So the cake pathway, as I mentioned earlier, will have an manually draw the pathway maps to understand the knowledge of the molecular interaction and the relations of networks with metabolism, genetic information, organism systems, human diseases, and also for the drug development. So try to retrieve, just call the pathway and then or you call the like a print like an think sub protocols and do this process. And once you do this process, it will ask you within we are trying to connect within a cake pathway. You can see here using our HTTP servers, the web, we are trying to connect a pathway and try to identify the number of compound list. So how many compounds you want to retrieve, it will see that specific gene. We are not using any docking technique. We are not using any pharmacophore techniques, nothing. We are just understanding only the genes. From the genes, we are trying to connect the compounds. How it is connecting, you can see here. From you look for what are the pathways, how many genes are expressed. From that gene, it will try to connect the compounds actually. So basically, the one is the most important is the KGRIST. KGRIST is a database of understanding the cell high-level functions utilizing a biological systems, especially the library KGRIST will try to take a data and generate the gene to pathway map and enrichment analysis and fold enrichment, and finally you end up with a molecular data sets. So KGRIST is the most important database that will have it playing a major role. So then from the cake, all the work is done. Now it moves to a CAS ID from the cake compound databases. It will try to pull from a National Institute of Standard Technological Library chemical book. It is used to search for the CAS registry numbers. And once it identifies, it looks for the 2D structures, 2D molecule, two-dimensional in a mol file format, and convert into smiles and standardize and prepare. Automatically it prepares and merge with the probe IDs and gene symbols. So wherever the specific gene, maybe a PTEN gene is expressing, what should be the drug has to be preferred. So it will do in this library analysis and retrieves your data. And this is the overall final power pipeline for this. So read your data from microarray, put for the fold change, select a property, extract a subset. Once you extract a subset of genes, add annotation and extract the individual genes. You can see the pass and then fail ports and go for the pathway analysis and make annotation. Whatever the genes are making annotations, those only will go into this and add the components to that and pick a compound range. You can also set the Lipinski's rule of 5 over here also and then make a CAS ID number and get the structures from the CAS ID and view your results. So to do this process, you might have been thinking how long this is going to take. It's just a fraction of 1 or 2 minutes. I'm going to show even demonstration also. So this is how the output will be there. Molecule, gene ID, and pathway ID, Cas number. Like this, you have a tabular columns. You can view the results. Now, let's see the demonstration and come back. Let me open and show you the pipeline palette. 1 minute. I'm trying to open the pipeline palette and coming to the share window. Sharing my screen again. Okay. So this is a pipeline palette window. You can see here components, protocols. If you have the components, you can see the enriched components. So if you want to type fold change, just pull the components. And if you want to look for an, for instance, I want an annotations. I want to tag a surface area or volumes or an protein-based tools, whatever I wanted. I can put a word like this. Or I want a viewer files. So we have these many viewers, clustering geo chart, clustering heat map analysis, dashboard analysis, HTML viewer, peptide viewer. So like this, we have a various viewers. So this will help you a lot to understand, to run a more in-depth knowledge in machine learning algorithms. So now I'm going to the protocol. Say for instance, I would like to go for an gene expressions. So I immediately have a inbuilt protocol. So I can either take a as such or I can clone like this. Control C, go to File, New Protocol window, Control V. If you want to make changes in your protocol, you can very well can do over here. So I will show here. So I'm running the protocol. Already it has been, I have taken the cell structure of an microarray analysis, going to introduce the fold ratios, factor 1 and factor 2, select a property, fold change, had extract the subset. If you double click and open, you can see it. And going back to the protocol, add annotations. If you'd like to add some other annotations also, you can try to add. Maybe I can add gene name or extract individual genes. So rename a genes and properties. Going back, get all pathways of Miami annotations, compounds, pick compound range, Cas ID, structures. Let's run this. You can see it in job menu. It is running in the job menu. So it is filtering. You can see the options over here. As it keeps reading on, automatically it gets into the pipeline. If you double click and open, there the pipeline is going on. That also you can see. Going back. Yeah, we got the results. So for this molecule, take map ID and genes. So it will display the genes and their respective molecule. So this is how we are trying to bridge our input of a gene expression data, right, to connect with the various pathways and finally retrieving your compound information. So that's an information about how to bridge your gene expression with your chemistry. Hope everyone enjoyed. And if you have any queries and anything need to be done, please connect us with an Altum inquiry at Altum Technologies or some kind of a technical query. Feel free to write us. So thank you everyone for watching us. Thank you. Okay. If you have any queries, you can put it in a chat. We are very happy to answer all your queries. Or you can also connect to us. Okay. Uh, I mean, good evening, Mr. Naga Pradesh. Your query is about what are the chemical components or used in RDNA technology? You mean basic RDNA technology, or could be a little bit specific, or which you want? Because all this are NGS technologies or microarray data analysis, RDNA recombinant technology. What technique you are looking for? You mean a PCR or some kind of what do you mean by chemical component? If you could let me know about it, that would be great. Okay, so hope you all enjoyed, and I believe that this will give you the more information to all. And if you have any queries also, uh, you can connect to a specific micro injection method. Okay, I will tell you in detail. Say, for instance, here what we are trying to use in this pipeline. You must have the output of any gene technology. Say, for instance, gene expression technology. Either your results of whatever you might be doing RDNA technology, whatever DNA technology that you're trying to use, that output must be there in a specific formats. You are using in a platform, which platform you are using, that specific platform alone can be taken over here. Using that platform, you can see the genes which are getting expressed, and through that, you can able to connect the pipeline. This is little bit specific into the technology, but what technology we are trying to explain over here is an output of your gene expression. Maybe you are looking for a microarray, or maybe you're looking for an RNA-seq or whole genome seq. That output can be taken, and you can connect to the this bridging technology. Okay, I hope Nagar Pradesh, if you have any queries, also write it to us. We will connect you back.